Hi, I'm Daniel and I'm in my Master Studies of Biology. This year I was part of something big, something called Igem.
I was always curious about how an own research project in the field of synthetic biology would be like.
I always liked solving problems and wanted to make tasks, even simple ones, simply effortless.
I wanted to think outside the box and get to know more about all the different facets a research project comprises.
We found all this and even more in Igem, the international genetically engineered machine competition. It is a way to get young scientists and engineers working together to modify present or develop new biological systems. This year, around 280 teams from 40 different countries took part in it. In April, 19 students of the RWTH Aachen University aimed to shape the future of bioeconomy. They started to work on their project, UMix, upcycling methanol into a universal carbon source. The use of oil is the main reason for climate change. If we want to get independent from oil, we need to find a new way to obtain fuels, plastics and currently oil-derived products. But nowadays, atmospheric CO2 can also be taken up by a technical process. It is then converted into the cheap and high-energy alcohol, methanol. Unfortunately, creating complex molecules for methanol is hard. Therefore, we are upcycling methanol into the universal carbon source, glycogen. This glycogen can then be used for the production of a great variety of goods. Thereby, we are going one step further into a fully sustainable and independent future. But how do we achieve the microbial conversion from methanol to glycogen?
The first step of our own research project was to engineer bacteria that are able to grow on and use methanol. Therefore, we used a synthetic biochemical pathway that was just published some months before, the methanol condensation cycle. The second step was to accumulate glycogen in the cells. As one of the first groups in Aachen, we used a version of the fancy new CRISPR-Cas genome editing approach that is suitable for E. coli cells. Using this editing method, we wrote Igem Aachen into the genome of our bacteria. A big part of our experiment design was performed on our computers. We used the newest software tools like Genius to plan the procedure. As a final result, we have developed various bacteria strains that are able to grow on methanol and produce glycogen. But we also needed an efficient way to analyze these bacteria.
So, we decided to build our own mini-scale bioreactor. We wanted to make biotechnology accessible to everyone. While developing the bioreactor, it was a challenge to use standard, inexpensive parts and customize them. Following the do-it-yourself principle, we constructed our own pumps, stirrers and online sensors. To enable easy use of the bioreactor, we developed the Master Control program. It is a software that allows users to visualize and control the operating parameters of the bioreactor. It basically handles everything from calibration to cultivation. Our bioreactor system is so simple that even schools can assemble and use it. But Igem is more than just science and engineering.
Managing, presenting and evaluating of your own project is a major part too. Since we care for project funding mostly on our own, we developed a fundraising strategy and got in contact with companies and academic institutions. We were also interested in the future of biotechnology in our everyday life. There are certain open labs in Europe which allow everyone to come in, learn and perform their own biological experiments. They are called community labs. Together with a Dutch and a German community lab, we addressed social, legal and biosafety aspects related to our and their work. All in all, it was a very exciting year for us. But what did we achieve?
I'm proud to tell you that this year all 19 members of our team went to the Igem Finals in Boston. We presented our project and were rewarded with a gold medal. In addition, we were also nominated for the best software tool. But on top of that, we outran all the other teams in our track and were honored with the Best Manufacturing Project Award.
To be part of such a huge project without having any prior practical experience makes me proud of my team and myself.
But there's also a different kind of joy in Igem. Seeing a 12-year-old boy telling his mom how much he fancies the bioreactor, it's simply amazing.
All this and much more is Igem. Do you also want to feel the Igem experience? So what are you waiting for?